So are you all able to see my PowerPoint presentation?
Yes, sir. Ay, wala pa, sir.
Ay, wait. Asa naman day? Ah, akong i-refresh ha. How about now? Can you see the slide?
Yes, sir. Makita na, sir.
Okay. So thank you everyone for the confirmation, and Abigail. For this afternoon session, we'll be talking about the bacterial cell structure. No? So basically, we have two parts for this topic. But first, let us start with our bacteria taxon. No? So later on, next week, we shall discuss the arterial cell structure. Okay. So for your bacteria, basically they have various or wide array or wide spectrum of cell shape and arrangement. No? But they have these particular groupings of shapes as to where we can eventually also group them together according to whether they are circular or run shape or even filamentous or curved. No? So when we try to look at the common shape and arrangement of our bacteria, we have this particular kanang sets no, of shape. So first one, we have what we call coccus. So your coccus bacteria are actually quite rounded, no, or spherical. Then you also have bacteria that are rod shaped, meaning these are your bacillus no, type bacteria. And then you also have the curved ones. For the curved ones, you can either have what you call a spirillum or a spirochete or a vibrio. So what's the difference between the three or the curved rods bacterial shape? So when you say spirillum, these bacteria are actually quite spiral, and the cell wall and the plasma membrane altogether are very rigid. So they are spiral and then they are not flexible at all. When you go to the spiral type of bacterial cell, which are quite flexible, so meaning when they try to move, they can bend the part of their cell. Now these type of spiral and flexible bacterial species are what you call spirochete. No? And then you have the very blunt rod-shaped bacteria which can either be in a comma shape. So these are now what you call vibrio. No? So, but all of these three types of shapes are actually quite grouped together generally as curved bacterial shape. No? So basically, the spiral are your coccus, the rod shape are your bacillus, and these elongated and flexible or rigid or comma shape are the curved rods. Now, there are also bacteria that are quite shaped that are or that cannot be, no, um, that cannot fit on in these particular categories to either spherical rod or curved rods. So naasan sila laing ilahang own shape, no, and these are now what you call pleomorphic bacterial shape. So there is no distinct or definite kanang characteristic shape for your particular bacteria. So aside from being spiral, aside from being rod shape or curved rod, if you can see other types of shape, then these are actually what you call pleomorphic. No? So na sila lain-lain na shape. kumbaga. So for example, you have palisade, no? So these are quite columnar, no, type of bacterial cell shape, and as aside from being columnar, they're actually grouped together or aggregate with one another. No? So pleomorphic, one particular example would be a palisade. Another type of bacterial cell is what you call a hypha, no, or hyphae, your mycelia or mycelium. In your hyphae, these are actually filamentous, no, bacterial shape. So when you say filamentous, these are very, very much elongated, very long, no, in terms of its kanang length, and these are actually um, made up of different aggregations of cells with a certain level of differentiation. No? So when you go to, for example, kanang mga cyanobacteria, no, your cyanobacteria, these are photosynthetic bacteria that are found in our aquatic environment. They have this type of bacterial cell, no, and some of these cyanobacteria, they do have this differentiated cell along the length of their hyphae. You call them as, for instance, heterocysts, no, and these heterocysts are actually functioning for nitrogen fixation. So basically, that's how they get and fix their nitrogen coming from the water, and then they are now able to photosynthesize and use this nitrogen for manufacturing their own food, no? As well as providing the macromolecules that are essential for them to create structural proteins. No? So, kailangan sudsud nila ang nitrogen. So, these are just kanang the common, no, bacterial shapes that we can eventually see under the microscope. But since wala man tayo sa laboratory, then you can just Google some of these examples, no, as you do with your exercise, laboratory exercise number 2. No? So our laboratory exercise number 2 are all about bacterial cells, no? Most especially under the kingdom Eubacteria. Okay, so again, we have coccus for spherical shape, bacillus for your rod shape. Then you also have curved rods, which could either be your spiral, no, or the comma shape. So under your spiral bacterial shape, you can have spirillum, and you also have the spirochete. And for your comma shape, you have vibrio, just like your vibrio cholerae. For another shape is your pleomorphic, meaning they do not have kanang definite characteristic shape. So aside from being spherical, being curved rod, being rod shape, so they have another type of shape. And then lastly, you have the filamentous bacterial cell, which you have the presence of hyphae and other differentiated cells, no, along its length. Now, aside from having our common bacterial shape, you also have what you call bacterial arrangement. So your arrangement is different from shape because the shape is actually the how the cell is kind of providing us a definite structuring because of the presence of its cell envelope. But this time around, when you say arrangement, it has something to do with the organization of the cell, no, with respect to the other independent bacterial cell. Now, in your arrangement, you can this is basically dependent on how they are arranged as well as the number of bacterial cells that are associated with one another. For example, if you only have one particular bacterial cell, then the prefix for your bacterial arrangement would be mono, no? So meaning mono 1. If there are 2, then you have diplo, no? If you have 4, then you have tetrad, no? If you have 8, you have sarcena. Okay, and the arrangement also is basically dependent on the actual shape of your bacterial cells. For example, if you have only one bacterial cell that is spherical, then you call it as monococcus. If you have two bacterial cells that are spherical, you have diplococcus, and so on and so forth. So you can also have monobacillus or diplobacillus. If you have bacterial cells that are arranged in chain, meaning one end of the bacterial cell is attached to the other end of another bacterial cell, then you call it as strepto. no? Strepto meaning chain. So they are forming a sequence of different independent bacterial cell. So if your chain is made up of spherical bacterial cell, then you call them as streptococcus, no, or streptococci. If they are made up or consisted of rod-shaped bacterial cell, then you can call them as streptobacilli, no, or streptobacillus, and so on. If you have, for example, a cluster, no, a grape-like cluster of bacterial shaped organisms, for example, a spherical bacterial cell, then you call them as staphylo. No? So staphylo meaning grape, and the arrangement of these bacterial cells are not in a certain way, but these are just irregular and random. So giklump ni sila. That's what you mean by staphylo. So staphylococci, staphylobacilli, and so on. So for your arrangement, therefore, the naming of your arrangement for your bacterial cell is based on the shape of your bacterial cell as well as how they are arranged, particularly in response to the other independent bacterial cell. Now, why do we have these different arrangement of bacterial cells? So it has something to do on the plane of cellular division, no, as your bacterial cell divides through asexual reproduction or through binary fission. So for example, there is only one plane of division, then that could possibly can lead to the diplococci or the streptococci type of cell arrangement, no, or diplococcus and streptococcus, no. If you have two different plane of divisions, then that means there are actually four particular cells that can be attached to one another. So basically, after binary fission, the independent daughter cells do not detach from one another. So if that's the case, if dili sila magbuwag, then there is now the possibility of having these different types of arrangement, no, bacterial cell arrangement. So again, if you have two plane of divisions, then that could lead to your tetrad. And if you have three particular plane of divisions, then that could lead to the formation of Sarcena, which is actually made up of 8 different bacterial cell. And of course, the Staphylo, which is an aggregate of more than 8 bacterial cell, which are uh, arranged randomly. No? So for your arrangement, you can have the monotype, the diplotype, the streptotype, the tetrad type, the Sarcena type, and the staphylotype. Okay, so you can eventually have a specific arrangement and cellular shape if you combine now the specific type of the bacterial cell shape as well as the bacterial cell arrangement. No? So the shape and arrangement of your bacterial cell are usually in the past, no, traditionally are used to name your bacterial cell. Now, that's why you have now what you call Staphylococcus aureus, no, meaning the arrangement and the cellular shape of the bacterium is actually spherical and they are arranged randomly in a grape-like manner. No, you can also have Streptococcus pneumoniae. No? So if you hear that particular name, then that would give you an idea that the bacteria is arranged in a chain-like manner, no, and the bacterial cell are also spherical. That's the name Streptococcus pneumoniae. No, Sarcina lutea is another bacterial species, no, as to where the Sarcina part of the genus of your bacteria suggests that it is made up of 8 clumps of bacterial, spherically shaped bacterial cell. No? So basically, if you have a background on the shape and the arrangement of your bacterial cell, then you will have a rough idea on the structuring, arrangement, and the shape as well of your bacterial species. No? Na as a name nila. Now, we ask ourselves, why is that there are several types of bacterial cell shape as well as that the size of your bacterial cell are very, very small, no? Approximately, the smallest would be about 1 micrometer and the biggest is around 600 micrometers. So, in ana ra kagamay ang inyong bacterial cell. No, very, very tiny. But then again, the question is, ganong tiny man sila, no? How does this particular adaptation in terms and with regards to their size, no, become so efficient for them, no? So it really the cell size of the bacterial cell has something to do with the surface area to volume ratio, no? So this is the indicator. This is an indicator for your bacterial cell that the nutrient acquisition and assimilation from the environment is actually quite efficient. No? So if you try to look at the surface area volume ratio, if there is an increase in the S over V ratio, then that would also mean that there is an efficiency in nutrient acquisition. No? Meaning, the greater the surface area and the lesser the volume of your bacterial cell, the greater the ratio becomes. No? So if you increase the cell size of your bacterial cell, then therefore the volume would also increase, therefore decreasing the SV ratio. So it has really something to do with the surface area and the volume. No? Why is that? If you have higher surface area than volume, efficient in nutrient acquisition. Because take note, the surface area of your bacterial cell, no, is made up of the cell envelope, and in your cell envelope you have there one particular layer in your cell envelope is your plasma membrane or the cell membrane, and on your cell membrane you have different types of proteins, and these proteins could act as transporters for your vital and necessary and molecules from the environment to be moved or transported inside the cell. And these particular molecules are actually essential for the survival and thrive and maintenance of your bacterial cell. So eventually, kung dako ka og surface area, meaning dako sa kag area nga pwede mo absorb sa mga nutrients nga present sa environment. No? So that is why when you have higher surface area and lesser volume, it could eventually create a smaller sized bacterial cell but highly efficient in nutrient acquisition. So that is why gagmay jud kaayo ang ato ang size sa bacterial cell. So it has really something to do with surface area to volume ratio. Now these are the different structures that we can eventually find in your bacterial cell. So basically there is still a degree of organization, no? Because your organization is one particular character of all living life forms. Now, without organization, dili siya ma-consider as a biotic component of the environment, no? Or a living thing eventually. So what are these kanang mga essential structures that we can find in your bacterial cell? First one is your plasma membrane. Now, let's look at our table here. Your plasma membrane or your cell membrane, no, is the very barrier that kanang separates the internal environment of your bacterial cell from the outside world. No? So your plasma membrane is actually very much important because it also serves as kanang a semi-permeable membrane. No? Meaning it allows some particular molecules from the environment to go in and the waste products inside your bacterial cell to go out. No? So I think you already know about the semi-permeability function of your cell membrane. No? So it doesn't allow all your kanang materials to pass through the plasma membrane. So mura siya security guard. No? Then just outside your plasma membrane is what you call a cell wall. No? So your cell wall here, since this is a bacteria, is made up of several glycoproteins, but the most conspicuous material that is present in your bacterial cell wall is in the form of peptidoglycan. No? So in your peptidoglycan, again, you have there the katong N-acetylmuronic acid, no? And then you also have your katong N-acetyl glucosamine, no? So nag ug nag. And then outside your cell wall, you also have the capsule. In your capsule, or you can also call it a slime layer. Now, this is the very layer that is resistant to phagocytosis. No? And it is also used to connect or attach your bacterial cell wall to any substrate, most especially host cells. No? So, mura siya na ay glue na pwede siya mo attach now sa inyong host cell. Altogether, your plasma membrane, your cell wall, your capsule or the same layer, they now comprise what you call the cell envelope. No? So ang cell envelope di ay is a complex structure made up of several layers. No? The plasma membrane, the cell wall, and the capsule or the slime layer. Okay? So within your cell envelope, then you can now see the various materials and the various structures within the cytoplasm of your bacterial cell. So for example, by the way, in your bacterial cell, diba, these are prokaryotic type of cells. So that would mean in your bacteria, they do not have the nucleus and any membrane-bound organelles. So wala sila tong nucleus, walay mitochondria, walay chloroplast, walay endoplasmic reticulum, both the rough and the smooth, walay Golgi apparatus, walay membrane-bound vesicles, no, wala tanan. So what do they have inside their cytoplasm is actually a specific region as to where they are protecting the genetic material of your bacteria, no, the chromosome as well as the plasmids, for example, and also the inclusions. No? So when you say inclusions, these are actually inorganic counterparts or inorganic materials that are present in your cytoplasm. So what are these inclusions? For example, they can be in the form of nutrients, no, the macro and the micronutrients. They can also be in the form of water, no, the buffer inside your cytoplasm, all non-living materials inside your bacterial cells. So these are what you call inclusions. No? Aside from the inclusions, again, you have the nucleoid, which is the specialized kanang place within the cytoplasm of your bacterial cell as to where you can find the genetic material of the bacterium. No? So your genetic material here is being protected from any other materials within the cytoplasm via specialized proteins. Now, you call them as nucleoid-associated proteins or the NAPs. Now, so later on, we will also have discussions on these particular proteins. You also have what you call periplasmic space. So in your periplasmic space, from the name itself, this is the space as to where you can find it in between the outer and the inner membranes of the cell envelope. No? So later on, we will also have the structure or kanang a figure that will represent our periplasmic space. No? What else? You also have flagella. No? So for your flagella, most of our bacterial species, they do move using flagellum, not cilium, no, or cilia. So most of our bacterial cells are being presented with numerous flagella for them to move from one location to the next or to displace themselves. Other flagella are used for by your bacterial cell to attach to a host cell. No? So daghan sad siya kanang plethora of functions and yung flagella. And then you also have what you call endospores. So for your endospores, these are the very survival features or characteristics of your bacterial cell. Maunang, kung for example, inyong initon ang certain material na agihapoy mutubo nga bacterial cell because your endospores are actually very highly protected kanang seed structure within your bacterial cell that can eventually endure very harsh environmental conditions. So this would allow your bacterial cell to eventually withstand no kanang mga overwintering or very low temperature, very high temperature. So if this particular harsh environmental condition would eventually pass through, mahuman na, then your bacterial cell could now be able to grow and develop again, no, via the endospores. So mo na sila ang atong patyon when we do sterilization process eventually. So when we sterilize our for example, agar or any glasswares in the microbiological laboratory, no, under 121 degrees Celsius, under 15 psi or uh, pressure per square inch, then we are actually kanang killing the endospore as well as the bacterial cell itself. Kung ato ramanggong i-boil ang inyuhang water, for example, no, boiling is kanang not a guarantee that we are actually killing all the bacteria, but at least we are killing some of pathogenic bacteria. But ang dili mamatay ani are those bacteria that can eventually create the endospore or can kanang unsa ni can develop the endospore. No, so ang endospore din na siya very very thick na cell wall or capsule na nagprotect sa inyuhang interior portion, which is viable when your environmental conditions tends to become more advantageous for them, no. What else? Unsa ang wala na to na discuss? Ribosomes, no. So ribosomes are essential for protein synthesis because these are the very structure inside your cells, both prokaryotic and eukaryotic, where new proteins are being manufactured every time. So without your ribosomes, you cannot create the cell eventually because try to remember, your cell is made up of proteins, diba? And your proteins are the building blocks of cells. So all of your cells are actually under the mercy of your ribosome because these are for protein synthesis, no, site for protein synthesis. You also have what you call gas vacuole. So these vacuoles are not eventually membrane bound because you don't have kanang double lipid bilayer in your bacterial cell, but rather the gas vacuole here are being coated or being made up of proteins themselves. So unlike the gas vacuole in eukaryotic organisms, which are made up of membrane-bound double lipid bilayer, no, these are not made up of these type of material, but rather they are just kanang made up of proteins, no, ang ilahang gas vacuole. What else? Pili or fimbriae. So these are actually cytoplasmic projections which are used by your kanang bacteria to attach themselves to another bacterial cell during conjugation processes. So what is conjugation? This is actually one type of what you call horizontal gene transfer, no? Wherein your bacterial cell can eventually kanang maintain kanang genetic variation, no. So they during conjugation process they can eventually exchange no genetic material. So mo nilang gamiton ang pili and fimbriae. These are like bridges no nga mo connect sa duha ka bacterial cell for them to allow genetic exchange, no? So conjugation is a way for them to maintain genetic variation. So I think these are just no some common features that are found in our bacterial cell. So again, you have the cell envelope, which is comprised by your plasma membrane, the cell wall, and the capsule or the slime layer. Then inside, you can now have all of your inclusions or the non-living materials, as well as the ribosomes, so the gas vacuole. You also have endospores and the nucleoid, and your cytoplasmic projections and kanang unsa ni nga mo project outside, for example your flagella, your fimbriae and pili. You can also see that one in your bacterial cell. So for your flagella for movement or attachment, and for your fimbriae and pili for attachment on other bacterial cell for genetic exchange. Now let's try to zoom in our focus on the bacterial plasma membrane. No? So again, your bacterial plasma membrane is the very structure that controls the material's movement going inside and going out of your bacterial cell. No? So security guard jud siya. Siya ang mobantay kung kinsa ang musod, kinsa ang pwede mo gawas, no? So that is what you call semi-permeability, no? So that's a character of our cell wall, not only in your bacterial cell, but it is also true with respect to our eukaryotic representatives, no? So here you can actually see that you have two different types of this is not cell wall, but this is actually looking into no, not cell wall. This is not plasma membrane, but you are actually looking into the cell wall, no? So here, if you try to look at our figure, in your gram-positive bacteria, you have only one particular layer of your um, plasma membrane. Okay, so your gram-positive bacteria, they do not have the inner and outer membrane. In comparison with your gram-negative bacteria, you have two plasma membrane. Now, you have now your outer membrane here, and you also have the inner membrane. Okay, so within these two membrane, you have what you call the cell wall, which is made up of your peptidoglycan. Same here in your gram-positive. But take note of the difference in the cell wall of your gram-positive and gram-negative bacteria. The cell wall in your gram-positive bacteria is quite thick compared to your gram-negative. That is why your gram-positive bacteria do not have the outer membrane, because this is now occupied by the very thick cell wall. As opposed to your gram-negative bacteria, no, in your gram-negative, since the cell wall or the peptidoglycan is kind of thin, so there is now an outer membrane that could also be a portion of a gram-negative bacterial cell wall. No? So the difference in terms of being gram-positive and gram-negative bacteria is how thick the peptidoglycan part of the cell wall is, no? Because when we try to do your gram staining, it has really something to do with the retention of the crystal violet dye within the cell wall of the bacteria. So if it does retain the dye, then that would mean that it has penetrated the peptidoglycan part of the cell wall. So meaning it does not have the outer membrane. So if that's the case, then your gram-positive bacteria would be colored or will be dyed as blue or violet. No? So your gram-positive bacteria are all blue or all violet, no, when you do the gram staining. However, when you have the gram-negative bacteria, since you have the presence of the outer membrane, your crystal violet or methylene blue dye cannot penetrate the outer membrane. So what would remain, therefore, in your peptidoglycan is the violet, no, not violet, but the pink dye, which is what we call a safranin or the counter stain, no, which would render your gram-negative bacteria colored pink. So, mao na siyang lain-lain silang color. It has something to do now with the exposure of the peptidoglycan to our dyes. No? So, kung masok gani siya sa methylene blue or the crystal violet, then the peptidoglycan will turn blue. Then that is a gram-positive bacteria. And that would also mean that the bacteria does not have the outer membrane that hinders the absorption of your crystal violet. Now, if there is the presence of outer membrane, so ma-wash off man ang imuhang crystal violet or methylene blue during kanang counter staining. No? So, in counter staining mo, butangan mo itong safranin, ang safranin rang mahibilin. So, mawa ang color blue, ang mahibilin would be the pink color. And that is an indication that your bacteria is gram-negative, meaning very gamay ang iyahang peptidoglycan layer sa cell wall, and there is a presence now of the outer membrane. Okay, so that's the main idea or main gist of having gram-positive and gram-negative bacteria. Now, in your plasma membrane, you have there a plethora of different types of proteins, no? And here you have what you call peripheral protein, and you also have integral proteins. So in your peripheral proteins, from the name itself, these are at the peripheral region of your cell membrane. So wala siya nag-traverse sa inyong lipid bilayer, no? So narin siya sa one end makita. Whereas you have also what you call integral proteins. So when you say integral membrane proteins, these are now the proteins that traverses both sides of your plasma membrane. So if you try to look at your plasma membrane, diba, it's lipid bilayer because you have two layers of lipids. And these particular layers are actually made up of amphipathic phospholipid um, um, organic molecules. No? When you say amphipathic, that means that there are two particular ends of your lipid or your phospholipid in this case that are quite opposing in terms of their kanang unsa ni affinity to water molecule, because one end of your amphipathic phospholipid is hydrophilic, meaning they are water loving, and the other end is hydrophobic, no, or non-polar, or they do not like your water molecules, no. So as they create the lipid bilayer of our plasma membrane, the outer portions now are the head portions of our phospholipid, which are hydrophilic, and the interior of our plasma membranes, these are now hydrophobic. No? So eventually, if you now have the head portions that are outside, facing outside your plasma membrane, then that would mean our plasma membrane can allow certain polar molecules to enter the bacterial cell or any type of cell. No? And for your non-polar materials, these are now hindered by, from entering our plasma membrane. But they can enter the plasma membrane if there are specialized proteins, just like your peripheral and integral proteins, that will act as transporters so that these non-polar materials could be eventually kanang transferred or moved towards the interior portion of the cell, no? Or going out. No? So that's how our plasma membrane works. No? Kanang it does not allow all material to pass through because of its innate characteristic of being amphipathic. No? Ang outer part is hydrophilic and the inner part is hydrophobic. Now, um, this particular lipid bilayer, no, you have here the the structuring of your phospholipid. So eventually, the head portion is made up of kanang polar, no, group of organic materials, and then it is linked to your glycerol with the presence of phosphate group. No? So eventually, for your glycerol, these are made up of two fatty acid chains, and this means now your tail end of your phospholipid as hydrophobic because you have fatty acids. These are lipids. No? So dili man nimo ma-mix ang water with oil, no, or lipids in this case. Now, um, I know that you are quite familiar with the what you call fluid mosaic model, no, which is actually the general concept or general depiction of our phospholipid bilayer. No? So when you say fluid mosaic model, fluid because you have now the presence of the phospholipid, the the lipid bilayer, no, which gives your plasma membrane its fluidic nature because of the hydrophilic and the hydrophobic parts of your phospholipid. And then the mosaic part is actually pertaining to the proteins that are present in your plasma membrane. No? So that's why fluid mosaic model. You have the phospholipid and you also have the presence of the proteins that are scattered around in your phospholipid bilayer. Okay. Now, in our bacterial cell, there is what you call a microdomain, which is actually an aggregation of several proteins that are now highly complex so that they can eventually move certain molecules from the environment going in, or they can also function as receptor for environmental stimuli for your bacterial cells to react. No? So it has something to do with their characteristic of being irritable, no, irritability. Now, this is quite different from fluid mosaic model because in our fluid mosaic model, it says that your integral membrane proteins are scattered in a kanang non-random way along the length of your plasma membrane. But this time around, in your bacterial plasma membrane, this is not the case because there are certain regions in your plasma membrane where proteins really aggregate to form protein complex. No? And this is now what you call microdomain. So you have there plethora of different types of proteins that are working together as a single unit. No? Most especially the presence of your flutilines. So your flutilines are your proteins that helps in the aggregation of these membrane proteins in bacterial plasma membrane. No? So they are highly specialized in terms of receiving external environmental factors or environmental stimuli rather, and they are also highly specialized in the transport of materials. So here, this depicts the very function of your plasma membrane as a semi-permeable membrane. No? So as you can see, your small nonpolar molecules such as your oxygen gas, your CO2, no, kanang mga gases that are essential for photosynthesis and respiration, they can eventually pass through the cell membrane or the plasma membrane quite easily. No? So they can pass with respect to your plasma membrane through what you call diffusion. No? So when you say diffusion, this is the movement of your materials from one region to another following concentration gradient. No? So in your diffusion, there is no kanang expenditure of energy because the movement is from a region of higher concentration to a region of lower concentration. Tama, no? And then when you go to diffusion, you have a specialist type, which is what you call osmosis, diba? What's the difference between diffusion and osmosis? Kasi kabalo. Yes, Abigail, sige.
Hey, sir. Can I be heard, yes, sir?
Yes, yes.
I've got answer. From what I remember, sir, the difference between diffusion and osmosis is that osmosis regularly talks about the movement of water across a semi-permeable membrane
Yeah.
from an area of high water to low water concentration, and diffusion, on the other hand, is I wouldn't say it's also water super. It's also from high concentration to low concentration surface.
Yes, no, precisely, Abigail. Thank you. So when you say osmosis, osmosis is a type of diffusion. No, so it's really kanang part of being a diffusion process. So your diffusion is the general term of the movement or net movement of materials from a region of high concentration to a region of low concentration. So when you say diffusion, it could either be you are kanang talking about the movement of the solvent water or the solute, no? For example, salts. So it's a general term. But when you are now referring specifically to the movement of water molecules from a region of high concentration to a region of lower concentration, then that is what you call osmosis, no? Through semi-permeable membrane. And in this case, we are now looking into our plasma membrane, no? So your small nonpolar molecules could eventually kanang pass our plasma membrane easily because of diffusion processes. And one of them would be osmosis for your water molecule. But there are also instances that your water molecules transport is regulated by your plasma membrane. Kaya kung dili siya ma-regulate, what would happen sa your plasma membrane? Di ba, muburot siya niya, mubuto. No? So to prevent that from happening, there are other ways of transporting your water molecules going in and going out. Now, for example, you have what you call protein channels that are named or called aquaporins. No? So these protein transporters are very specific for the transport of water molecules. No? So lahat yung mga water molecules nga i-regulate kung sila dili tuyang o pasulod or pagawas ba, because water is quite essential in the turgidity as well as the structure, no, and maintaining shape of your cell, most especially for our unicellular bacterial organisms, no, kay isa ra gud ilahang cell, kung magugma na ilang cell di patay jud sila entirely, whereas when you go to multicellular daghan man tag cells, so if the kanang several cells kay mamatay lang, but we still have other cells to replace the dead cells, no? So eventually water regulation is very important when it comes to bacterial cell. And take note, water is one essential requirement for your bacterial cell to survive, no? Since without your water medium they cannot eventually uptake or they cannot acquire essential nutrients and materials from their environment. Okay. Your large uncharged polar molecules, these are now being rejected by your plasma membrane because, no, di ba, kana inyuhang plasma membrane, these are highly compact when it comes to their arrangement. So, large na mga molecules, they are not allowed to pass through, but there are certain ways or mechanisms that they can be allowed to pass through. For example, with the presence of protein channels or protein carriers. So these are protein transporters that will allow your large molecules as is in their structuring to be transported towards the inside portion of the cell. Or they can also be dismantled, no, kasi kung dako na kayo sila, very much complex, they can be dismantled, and the dismantled portions or parts will be transported going inside, and then they would eventually build the complex molecule inside again to form that particular complex molecule, no? So pwede nila gubon niya transfer dito ang kanang mga parts o niya i-reassemble na sa nila again sa sulod. Your ions are actually charged kanang particles, charged materials. These are never allowed, no, through simple diffusion alone. So you can only kanang transport ions or charged particles across your plasma membrane with the use of channels or what you call pumps, no? Kanang sodium potassium pumps, for example, no? So kay charged man ni sila, and take note, your phospholipid layer is amphiphytic, no? So naay part nga hydrophobic and hydrophilic, so they would eventually kanang repel the movement of your charged particles. Okay? Another kanang purpose of your plasma membrane is not only for kanang semi-permeability, but also it has something to do with respiration and photosynthesis, no? Di ba? You ask yourself nga, ang bacterial cell, some can photosynthesize, and most of all, no, most of them are actually able to respire, no, cellular respiration, for them to create their ATP or the adenosine triphosphate molecule for them to do their work, no, or for them to do cellular activities, because ang atong ATP kay mo man na siya ang ato ang kanang energy monetary unit, di ba? So you can ask yourself nga, if they can eventually do photosynthesis and cellular respiration, and your bacterial cell does not have any mitochondria and chloroplast, then how are they able to do these particular processes, no? So mo nang pangutana, that is kanang unsa ni being adapted, no, gikuha na siya nga mga functions, and then that function now falls on the plasma membrane of a bacterial cell. So in your bacterial plasma membrane, you can actually see there a convolution or a convoluted part of the plasma membrane where there are networks of infoldings and invaginations to increase surface area. No? So again, when you have increased surface area, mas increased po ang inyuhang efficiency in terms of chemical reaction, in terms of absorption of nutrients. No? In this case, the convolution of the plasma membrane is what we call the mesosome. And the mesosomes now are the very structure in your plasma membrane that will enable your bacteria to respire and manufacture their own food. No? So even though your bacteria does not have the kind of chloroplast, which contains the chlorophyll pigment, and even though your bacterial cell doesn't have the mitochondria, but they have these particular structures, mesosomes, on their plasma membrane. No? So try to imagine now the structure of your mitochondria. Di ba you have cellular matrix inside your mitochondria na kay enfoldings, no? Kanang naa kay inner and outer membrane. That's just the same and synonymous with the mesosome, no? Di ba mura sad siya kanang maze na ay mga convolution just like your mitochondria. And just like your chloroplast in your mesosomes, they can also harbor the green pigment chlorophyll. No? If the bacterial cell wall do have the presence of the chlorophyll. So, dili po sila makita. So, not only the plasma membrane of your bacteria is kanang functioning for the movement and transport of materials, but also they are now functioning for the production of food and also the maintenance of cellular activities through the production of ATP molecules, no, through the process of aerobic respiration. So that's kanang very important to take note, na wala man sila chloroplast, wala po sila mitochondria, but they do have the mesosome that eventually kanang do these kanang cellular processes. Then, speaking of surface area, which heightens or increase the efficiency of absorption of our nutrients, no, you have here kanang certain types of kanang materials that are quite needed or badly needed by your bacterial cells so that they can maintain and sustain themselves and they can survive, no, in their respective environments. Now, you have two types of kanang nutrients. No, you can have macronutrient or micronutrient. It depends on the quantity or amount of the nutrients that is needed by your bacterial cell. No? So if the quantity is quite large, no, the needed quantity is large, then these are now what you call macronutrients. So your macronutrients can be in the form of oxygen, hydrogen, carbon, nitrogen, sulfur, and phosphorus. So these are just kanang common elements po na needed po sa ato ang eukaryotic cells. No? So eventually all of these are macronutrients for both prokaryotic and eukaryotic cell. You also have what we call the micronutrients, no, or the trace elements that are needed in your bacterial cells in very small or tiny amount. No? So, example for these micronutrients, you have kind of copper, cobalt, manganese, no, zinc, and nickel. But some kind of microorganisms or bacteria, no, are able to synthesize kanang organic molecules that they need from these macronutrients and micronutrients or kanang mga elements. No? However, some microbes are unable to synthesize certain molecules needed for their survival and maintenance. And these molecules that your bacteria were not able to kanang synthesize or manufacture are what we call the growth factors. And they must be obtained, no, by the bacteria from their environment. So, kailangan din niya kuhaon, i-acquire, and i-assimilate from their respective surroundings. So, these growth factors rather, can be in three forms. So, you can have the amino acids, then you also have the purines and pyrimidines, no, which are actually the nucleotide bases in our genetic material. And then you also have the vitamins. Okay?
From scratch pa din. So, di may
In
kapangay sa mga
terms of nutrient uptake in your bacteria, these are the three highlights, no, or kanang important kanang concepts to take note. No? First one is that kanang your bacteria can only take kanang nutrients from the environment that are dissolved in water. So again, it is very important kanang for your bacteria to
Pasok ko tuwa
be
mag-ayo kay
in
di
an
ba kadili mo.
environment where they are
Kung matako
immersed
sa
in
iyo,
water
matakulas
because
ayos
they
na
cannot
hapit.
easily acquire these nutrients if these are not dissolved in water. So maunang kung naa
Sis
kay
kami din.
food, di ba? Ang kanang mga basa nga food, usually dali ra silang kanang ma-spoil, no?
Nakoy klase.
Kay naa man siya
10:30 ba?
tubig. No?
Pagod ni naon
Whereas
ko.
kung imong indicate ang food ug kaayo, dugay sad siyang madaot. That is why your if you have kanang moist nga mga foods, daghang tubig, mas maka acquire man gud og nutrients sa inyong bacteria in these types of environments, and therefore their growth and development would also kanang quite be exponential. No? So mas paspas pud silang motubo. Therefore, mas paspas mas spoil ang food, no? So that's quite important to take note han. So dili sila maka-acquire of nutrients from the environment if these nutrients or materials are not dissolved in water. No? Second, kanang bacterial uptake of nutrients, no, are highly specific. So one particular mechanism is actually kanang kanang one uptake mechanism is for one type of material or substance, no? So only necessary substances are acquired. So therefore ang kanang mga waste products, mga other substances in the environment which your bacterial cell will not eventually use, dili na nila kuhaon. So kanang normally dili sila mo bulk acquire from the environment. They just acquire what is necessary for them to survive, no? Kay if you try to imagine again, your bacterial cell is very small. So kung mo bulk transfer sila sa materials from the environment, asa man nila ibutang sa ilahang sulod, no? Nga gamay ra man ang space. So to kanang prevent it from happening, ang katulad yung unsa ilahang kanang needed, ang ilahang pong i-acquire from the environment, no? Next and finally, no, your bacteria are also able to transport nutrients into the cell even against concentration gradient. So aside from having the passive transport, which is the diffusion and osmotic kanang movement of your materials, your bacteria, they have ample amount of active transport for them to really get those necessities, no, necessary nutrients. Kay kung katumbok manggod nga mechanism of transport ilahang gamiton, which is eventually your diffusion, your facilitated diffusion and osmosis, for example, daghan kaayo ang nga dili nila gamiton ang makasulod. So that's why, since it's very specific sa ilahang mechanism that are kanang pinpointing to a particular material, then they also have ample kind of mechanisms of active transport na ilahang gigamit to really acquire these necessary substances, no? And when we try to look at now the material transport for our cell, so you have basically your passive and active transport, no, diba? So in your passive, the main difference in your passive and active, no, just a refresher, is that materials tend to move from a region of high concentration to a region of low concentration. So if it's from high to low, then that's passive because the movement is through the concentration gradient. But if it's against concentration gradient, meaning the movement is from low to high, no, concentration, then that is what you call active transport. So mura siya kanang maglakaw ka o padong bukid or pa no, or from the top of the mountain going down, dili siya kapoy, dili ka kailangan mo spend of too much energy because you are going with the gravitational pull of the earth, no? So you're not going against gravity. But if you are moving from lowland areas going to the top of the mountain, mas mo exert ka og force because you are going against the gravitational pull, no? Same here. When you are passive, then you don't need to spend energy just to move your materials from a region of high concentration to a region of low concentration. Your active transport, you need to spend form of ATP for you to be able to transfer your materials from a region of low concentration to a region of high concentration. So eventually, your body will use a mix of your passive and active transport, but most primarily will be using more passive compared to transport. No? So under your passive, for instance, you have what you call simple diffusion or the net movement of your materials. Then you also have the osmosis if you are referring to the movement of water. And then you have what you call facilitated diffusion as to where you now have protein transporters that have transfer of your materials. No? So all of them, walay expenditure of energy. When you go to your active transport, this can be divided into two types. No? So now your pumps, no? And then you also have the physical diffusive transport. So under the pump, these are your primary active transport and your secondary active transport. No? So the primary and secondary active transport, these are the primary transport mechanisms that your bacterial cell are actually using. No? For the vesicle, more or less, this is your cells, no? So you now have your exocytosis and endocytosis. So when you say exocytosis, there is a movement of your materials from the inside of the cell going out. No? So in paglabay o mga waste material. When you have endocytosis, endo meaning in, so the movement of your material is from the outside going in. And in your endocytosis, you can have what we call pinocytosis. You have also phagocytosis and receptor-mediated endocytosis. Now, your pinocytosis is what you also call cell drinking, no? So meaning what is being engulfed by your cell are actually kanang liquid material, no? And in your phagocytosis, this is food or kanang cell eating because in your phagocytosis, the material that is being engulfed by the cell is of solid, no, material. Now let's move to the very mechanism of transport in your bacterial cell, no? Oh yes, this is a depiction of our passive diffusion, no, or passive transport. So again, the movement is from high concentration to low concentration, no? So you're not going against concentration gradient. That's why when you have mga hypotonic solutions, hypertonic solutions, it has something to do with the concentration of water inside or outside the cell. So kung ang water ninyo mas daghan sa gawas, that is what you call hypotonic solution. And then your water molecule would tend to move going inside, no, the cell. And if you have hypertonic solution, meaning mas gamay ra ang water sa outside, then the water inside the cell would eventually move out. No? So that's passive, no, transport or passive diffusion. Now you also have your facilitated diffusion, no, that involves several transport proteins, and you have two major types. Now you can call them as channels, meaning these are pores present in your plasma membrane, and you also have carriers. Your carriers are excuse me, your carriers are also membrane proteins that are substrate specific. So meaning these particular proteins only carry or transfer or transport materials that are specific for their receptors and nothing else more. No? So your carriers are highly specific compared to your channels or the pores. No? So if your movement does not require an energy, but it requires the help of your transport proteins for them to move across the plasma membrane, then that is what you call facilitated diffusion. Also, in your facilitated diffusion, a transport protein will help move substances in the direction of decreasing concentration. No metabolic energy is required. Okay, and your facilitated diffusion, again, as I've said, no, is not an important uptake mechanism for your bacterial cells because the uptake of your kanang nutrients from the environment is highly specific for your bacterial cell. Now, what were no kanang being used by your bacterial cell in terms of getting or acquiring nutrients from the environment are mostly in the form of active transport. No? So there are several systems in your bacterial cell that will eventually uptake or acquire a certain nutrient via a certain protein. No? So here, kanang you can see that there is involvement of several proteins in your plasma membrane, and these are integral proteins because if you try to look at our kanang figure, it traverses no both ends of our amphipathic kanang phospholipid. So these are eventually kanang integral proteins. So your integral proteins can be protein transporters no, or carriers. Now, in your kanang protein carriers, they could either be kanang a uniporter or a co-transporter. When you say uniporter, from the name itself, only one particular material is being transported at a given time. And the transportation is in one direction only. No? So that's uniporter. But if it's co-transporter, then it could either be a symporter or an antiporter. Symporter meaning, from the name itself, sim, there would be movement of two different molecules in the same direction. No? So they are kanang sympatric no, or equal in terms of kanang movement and in terms of the direction. But when you say antiporter, no, antiporter, the movement is kanang of two different molecules, but the directional movement is opposing one another. So one going in and the other going out, just like your sodium potassium pump. No? So your protein carriers, no, these are kanang the types of protein carriers of your integral membrane proteins, which has something to do with how many unique materials they can facilitate in the transfer and the directional movement of the transfer. So uniporter or co-transporters. If they are co-transporter, then they could either be symporter or antiporter. Now, in your kanang bacterial active transport, you can either have the primary or the secondary active transport. No? So here, this is an example of your primary active transport system, which has something to do with what you call the ABC transporters or the ATP binding cassette transporters. No? So in your ATP binding cassette transporters, you have there one particular end here, which is the receptor or the solute binding part of your protein. And at the other end is the ATP binding domain or region of your protein. So the solute binding protein, this is the receptor for the specific nutrient that is needed by your bacterial cell to be transported going in. But at the other end, this is where your ATP molecule now will attach to your protein. As your ATP attached to your protein, take note that there would be hydrolysis of your ATP molecule. So therefore, from adenosine triphosphate, it will now release one phosphate group for it to become adenosine diphosphate. So duha na lang iyahang phosphate group sa iyahang molecular structuring. And if that happens, if your ATP hydrolysis happens, then your protein, the other end of the protein will now open up to allow the movement or the transfer of your kanang solute or nutrient from the outside to pass through the cell membrane. So this is what you call primary active transport system with respect to the ATP binding cassette transporters. But take note, if you try to look at the the kanang unsa ni the number of unique material and the directional movement of the material. There is only one type of material, no, at a given time that is being accommodated by your primary active transport, and there is also one directional movement of your solute or your nutrient. So here in your primary active transport, these now kanang unsa ni these are the very kanang active transport that usually associates with transporters that are uniporter. So usara ka material and usara sad ka direction of movement. That's primary active transport. Then you also have your secondary active transports. No? So here, the transporters in this type of mechanism are either symporters or antiporters. No? So if you try to look at our diagram, no? So for your sodium potassium pump, there are actually ions. No? These ions are actually charged, so therefore they cannot simply diffuse through your plasma membrane. No? So they eventually move towards or can also move outside, no, your plasma membrane. But if they are moving together, then that is a symporter, no, type of transport. And if they are moving together but in different directions, then that is what you call a antiporter type of transport. No? So, kanang sayon-sayon na siyang tanawon. And inyuhang i-take note rin jud is how many type of nutrients and kanang the directional movement of the transfer of nutrients. Now, your bacterial cell can also transfer nutrients in bulk. But then again, they do not simply use this one kanang normally or in common situations. No? So group translocation is a type of active transport in which bacterial cell transport organic molecules while modifying them. So this time, no, if you are having bulk transport or transport of kanang complex molecules from the outside going in, since wala man kay dako nga pore in your cell membrane, then they have to be kanang dismantled and then reassembled as the parts are kanang transferred into the interior portion of the bacterial cell. No? So, one of the best known group of translocation system is what you call the phosphoenolpyruvate, no, the sugar phosphotransferase transferase system, or the PTS. And in your PTS transport, there's a variety of sugars, no, that can eventually relate them and using the PEP as the phosphate donor. So, kuhaon sa PEP ang inyong katong phosphate such that mabungkag ang inyong complex molecule, and then that complex molecule will be reassembled as they are moving along towards the interior portion of the bacterial cell. You also have what you call the kanang siderophores, no, which are actually secretions of your bacterial cell that are highly specific with iron. No? So they bind with ferric iron that enables the accumulation of iron within the bacterial cell. So again, no, these are quite highly specific. So this time your siderophores are eventually specific for iron alone. No? So this is how your bacterial cell unsa ni kanang triggers the movement of the specific iron ions coming from the environment going in. No? And here, the iron is kanang unsa ni very much vital for our bacterial cell because there are more than one route of iron uptake that could be easily used to ensure adequate supply. No? So basically, since iron is quite kanang hard for your bacterial cell to get from the environment, they need to have different ways of acquiring iron, and only and siderophores are only one mechanism for them to uptake the iron coming from the environment. But what we are really trying to look here is that your bacteria, since they are quite small, then they really need to be quite strategic in their way of getting materials from the outside environment to the inside of their cell. No? Because if they do not become highly strategic, what would happen? Their storage, no, is quite limited. So therefore, excessive acquisition of nutrients coming from the environment could lead to imbalances in terms of their cytoplasmic contents or inclusion. And imbalances could either result into imbalances in osmoregulation, for example, which could can influence the shape of your bacteria. And what would happen if the shape of your bacteria would be damaged or destroyed? Then there would be lesser and lesser surface area that could accommodate the transfer of nutrients from the environment. So that could also lead to disruptions in their metabolic activities and therefore death, no, eventually. No? So isa raman sila ka-cell, they are unicellular, so therefore they need to be very, very strategic. No? So kailangan, this is also a testament that your bacterial cell is
Makita na
kanang
resemblance si
like
Chirog si
a
Kubiko.
human individual. No? They can eventually think
No?
and they can eventually adapt,
Ano ba?
no, to a changing environment. I think I'll just stop here, no, for your bacterial cell structure. Time check, it's 5:38. Okay. So, this is your assignment for this particular topic. No, it has something to do with the cell wall of your bacteria.
Nga,
If you have
Seven na
noticed,
yung
no,
assignment
during
karon,
our
no?
discussion, wala kay ko nag-use sa term nga prokaryotic, no? Because on the advent of our genetic, genomic, and biochemical analysis, we really know that your kanang archaea and bacteria um, are not on the same phylogenetic taxa or group. No, separate jud ang inyong bacteria and archaea. And thus, I would kanang strongly not use the prokaryote, no, during our discussion, but we have to specify whether we are talking about archaea and bacteria. And this time, we are talking about bacteria. No? So now, for your assignment, now for number 1, why might a microbe have more than one uptake system for certain substrates? I think you already have an idea regarding question number 1, but you just need to expound that one. No? Number 2, how does the difference in bacterial cell wall, meaning being gram-positive and being gram-negative, affects the bacteria's resistance to antimicrobial drugs, no, or antibacterial drugs? Number 3, how does the difference in bacterial cell wall, again, being gram-positive or being gram-negative, influence the antibacterial drug selection and administration? Okay, lain naman silang cell wall composition and structuring and arrangement. So is there any influence when it comes to antibacterial drug selection and administration? And number 4, what are the clinical implications of having different bacterial cell wall structure considering a patient has a bacterial infection? No? And how does this pose challenges to our health sector? And what are these challenges? No? Deadline for this one is next week, no, Wednesday. And I already also scanned your assignment number 1 and number 2. So majority of you has already submitted your assignments. No, so we will have 3 deadlines next week. Your assignment number 3, laboratory exercise 1, and laboratory exercise 2. Speaking of laboratory exercise, so let me share to you our laboratory exercise 2 for. Okay. So our exercise, laboratory exercise 2, this is all about the kingdom Eubacteria. No? So basically, this is the first portion of our kanang discussion this afternoon. So here you have several kanang bacterial species. So what we will do is, again, i-google na lang ninyo Okay, wala pa ta sa laboratory. And then, take note of the shape and the arrangement of these bacterial species. Okay, so some of them might be quite easy for us to detect the shape and the arrangement because of the name, no? Kay Spirulum, Vibrio, Staphylococcus, but some might be quite difficult. No? So try to search for the structure, shape, and arrangement of these sample bacterial species. Now, you also have here kanang listing of the cellular shape and arrangement. So the main purpose of this exercise is for you to um, identify the shape and the arrangement of kanang representative bacterial cells by looking into Google Google pictures and then creating a tabulated form concerning these particular information. So shape, arrangement, disease, cause if they are pathogenic, and the taxonomy classification. So you have phylum there, class, order, and family. No? So we also have several guide questions here. So kamo na lay basa. And for this one, we shall have the
Kaso, alam mo, ipakita sa Facebook. Pictures. Milo. Hmm? Nawa na sa sir, gibitray na sa siya, tagay, good na siya internet good. Mga sige na siya gawa. Sa ilaw nga ata niya, may siya nga, data ra daw yung gamit, na. Tagay niyo wifi si sir. Tagay niyo wifi si sir.
Wala na naka-andon sa kuwan eh. Online setup.
Naning ra ba kayo na siya, taga F2F na mo.
Wala na C4 sa Eugene Island.
Go. Si Forest.
Biya to, C4 din nga na to.
Kako na. Si Forest.
Kailangan na tayo.
Graveyard. Wala na dyan si sir. Bye guys. Bye guys. Annyeong. Pwede naman malive.